And so our research group, and I'll talk a little bit more about our team in a second, is really focused on looking at the environmental factors that are associated with recreational beach water in Canada. And so Binyam and I are going to go through some of our research that we've been conducting in the past few years, and we're excited to share with you today. And so swimming at public beaches is an increasingly popular activity in Canada. And it became very popular, even more so during the pandemic, where we saw record numbers of people attending beaches as people tried to really, stay local and explore the outdoors in local settings. And so we saw numbers really surge in the past few years. And it seems that popularity has really kind of continued on as we move more towards the later and post-pandemic phase. And so there's lots of recreational activities that are quite popular that include kayaking, paddleboarding, water skiing, and canoeing. And so there's lots of ways that people can be exposed to water in addition to actually being in the water. And so we're going to go a little bit over our background. I'll talk a little about our research team, and we'll talk to you about some of the research projects we have going on and some of the results that we have to date. And then talk a little bit about where we're going next, because we hope to continue our research and allow some time, hopefully, for some questions from people. And so, as I mentioned, we have tons of beaches across Canada, all the way from the West Coast marine beaches to the East Coast, and lots of inland beaches, lots of lakes. So we're lucky to have both marine and freshwater beaches across Canada. So understanding the water quality is quite important because with lots of beaches, we have lots of people accessing them, and it becomes an important from a public health perspective. And so poor recreational water quality represents a risk of acute gastrointestinal illness for people who engage in recreational water activities, but it's highly undetected and underreported. And this is unfortunate because the risk is higher in children between the ages of 0 and 10. And so we know there's lots of different etiologic agents that are responsible for this infection. We know that there's E. coli, but there's also Enterococcus, Salmonella, Campylobacter, Giardia, Cryptosporidium that are involved with acute gastrointestinal illness. But beach managers specifically monitor for the presence of E. coli in freshwater settings and often Enterococcus in marine settings. And these are fecal indicator bacteria that indicate the presence of other pathogens in the water. What's also important to note is that environmental factors are highly involved with affecting water quality levels. And so our research aims to understand how these environmental factors are associated with beach water quality. And so our team is comprised of public health researchers at Toronto Metropolitan University, obviously in Toronto, and we're specialized in epidemiology. Our team also has specialization in burden of waterborne infection and advanced analytic methods. And we are part of a collaborative group. We work in collaborations with participating public health units to generate knowledge of Canadian beaches, not just freshwater, but also marine beaches, and inform current beach monitoring programs. And so our team is led by Drs. Ian Young and Jordan Teston at the university, who are both associate professors. And we have several projects that are ongoing right now. We have we're going to talk to you primarily about today, which is looking at the environmental predictors. But we also have a beach cohort study that's ongoing, and we're going to be looking at algal blooms too. And I'll briefly mention those at the end, just out of anybody who might be interested in the work we're doing. And so fecal indicator bacteria tell us that there is presence of fecal material in the water. And E. coli is E. coli concentration is what's used primarily for freshwater, recreational water, and Enterococcus for marine waters, as I mentioned before. But the focus of our research is specifically in E. coli. So it's not that, we don't think Enterococcus is important, but we are going to be focusing today, and everything we present will be looking at how environmental predictors affect E. coli concentration specifically. And so our research is based primarily on the 2012 guidelines for Canadian recreational water quality, which set a threshold of 400 coliforming units per 100 mils, a single max sample, or a geometric mean of 200 colony-forming units per 100 milliliters. There is a new addition that's meant to replace these current thresholds. It's going to suggest that a single sample max of 235 CFUs per 100 mils will be the new standard, but our research was designed on the current 2012, even though I understand that a lot of public health units might be moving into the new guidelines soon, if not already. And so in Canada, beaches are regularly monitored by public health authorities through water sampling and beach assessments during the summer season. And recreational water quality falls under provincial and territorial jurisdiction. And these sample results are used to inform risk management and communication decisions. And this includes any beach postings or closures to reduce risk on the public. The frequency of this monitoring varies and sometimes quite greatly across jurisdictions and beaches. And so the public health units, as many of you know, determine this by various factors. It could be by popularity, historical results, or other political decisions. Unfortunately, there's limitations to the current monitoring program because culture-based methods can take 18 to 24 hours to process. And so the decisions that we're making about beach posting status are really based on the previous day's E. coli concentration. And so these delays can result in risk to swimmers, as we now know the environmental conditions can affect water quality in mere hours. And so understanding these environmental factors that are associated with water quality can help us inform beach monitoring programs. And so when I keep talking about environmental factors, what do I mean? Well, there's rainfall, which is commonly cited in the literature. We have air temperature, water temperature. We can have stream discharge, wave height, wind speed, ultraviolet radiation, turbidity, salinity in marine beaches, and tides in marine beaches. So there's several environmental factors that can be associated with water quality in our beaches. And environmental factors become more and more important as we face climate change. Now, here we see data from Toronto and Niagara region. So this Toronto data is going all the way back to like the mid-early 1800s up until 2020 approximately. And for Niagara, it's early 1900s until also the about 2021. And we can see an upward trend in our temperatures. And so really understanding our environment becomes increasingly important as well as we deal with climate change and the importance that's going to have on water quality around the world in general. And so our research objectives were twofold, primarily for this specific project today, was first to determine the climate and weather-related factors associated with higher levels of E. coli in public bathing beaches over time. And then second, from that information that we learned was to develop predictive models. And we're going to use this, do this by using a novel approach, which is called Bayesian network. It's a type of AI for selected beaches to assist public health authorities in risk management decisions. And so I'll start with the first objective, which was to determine the climate weather-related factors. And so we worked with 4 regions across Canada. We worked with Toronto. Toronto's an interesting area because it's the largest city in Canada, and allowed us the opportunity to study beaches in an urban setting in a Great Lake. So Lake Ontario, and the beaches are all located essentially right in front of the city. So very urbanized area, very urbanized beaches. So an interesting opportunity there. Niagara region was our next region, also very important because it's one of the most popular tourist areas in Canada. Large geographic areas spanning two Great Lakes. So we have Lake Ontario and Lake Erie. We also explored Manitoba, Lake Winnipeg, one of the largest freshwater lakes in Canada as well. And give us opportunity to explore another freshwater lake outside of our Great Lakes. And then we have Vancouver, also one of the largest cities in Canada, also coastal urban beaches, and opportunity to explore marine beaches. So 2 very interesting things that we can explore there. Another urban setting, but also marine setting. And so when we think about environmental variables, I just want everyone to think about the way that we've sort of designed our research is that we really think about environmental exposures preceding E. coli concentration. So say we're looking at today's E. coli concentration, we know that perhaps what happened in the previous day or the previous 48 hours is what's important. So whether it rained, for example, in the last 48 hours. And so a lot of our variables, we made decisions based on the literature in terms of whether to set it as a same day exposure or previous day. So for rain, we looked at cumulative precipitation in the last 48 hours. And for UV index, air temperature, wave height, and previous day coli, obviously uses previous day data. And then for things like bird counts and shore wave height and turbidity and stream discharge, we looked at same day. So what were the current conditions and how do they affect that day's E. coli concentration? And so Toronto, as I mentioned, there are 11 beaches on Lake Ontario. We included 10 of these in our study. And as you can see, they're essentially a big cluster right around the core of the city. And Niagara, as I also mentioned, spans 2 Great Lakes. And we looked at 2 beaches in Lake Ontario, one that ended up in the final analysis, and 6 beaches on Lake Erie. And so when we look at environmental factors, it's not just as simple as this diagram right here, where each environmental factor is just essentially, we're looking at it independently and how it has a relationship with E. coli concentration. It's not as simple as that, because the environment is an interplay. So, you know, air temperature, wind can affect wave height, which can then affect turbidity in the water as there's movement in the water. Rainfall also can stir up the water. So we know that the variables, there's interplay between them, there's association between them. And we need to think about that when we do, as epidemiologists, when we do our analysis. And so this is a very overall, the overall results of our path analysis for Niagara. It's a very specific type of methodology that's used to allow us to look at the relationships between variables. And it really allowed us to identify some trends across the region. Number one, that turbidity, as you can see there, the yellow icon there, was a very important variable across all the beaches in the Niagara. That if the water is turbid, it was very strongly associated with increased E. coli levels. And then a lot of the variables themselves were associated with turbidity, such as wave height was a very strong predictor of turbidity levels. So we're able to sort of identify which were the strongest variables in turbidity. I'd say was a big takeaway here. I'm not going to go into the details of that. We did present this research 2 years ago at our last NCCEH seminar. But in terms of our key findings, we confirmed some relationships that we already suspected or have been said in the literature. And we found that that beach-specific model, so the one I showed you was an overall model, but we found that there were some general trends. And as I said, turbidity was found to be a very important variable or mediator for the indirect effect of environmental predictors overall, and in all beach models across Niagara. And if you were interested in reading more about this, our results are published in the Journal of Epidemiology and Infection, where we specifically go into the methods of this analysis and the specific results. And if anybody is interested in looking at the beach level models as well, that essentially we confirmed some of what the public health unit and us as well suspected would be the important variables. We also conducted a Toronto and Niagara analysis to kind of not necessarily compare the 2 regions, but perhaps understand if there were some general trends going on. And we did find some trends, again, confirmed to what we sort of believed as hypothesized and what the stated in the literature. And so 24-hour air temperature, the higher the air temperature was associated with increased E. coli levels, as well as increased water levels. Previous day E. coli, which is, as I mentioned before, what is currently used in monitoring in public health units in terms of decision-making for risk management and beach closures, was a very strong predictor as well. So that's good to hear that it's definitely a good predictor of E. coli levels and wave height the previous day. Interestingly, but not surprisingly, UV index has a negative relationship with E. coli. So higher UV radiation means less E. coli concentration, which makes sense given that it can deactivate the DNA of the bacteria. And so we did find some differences between the 2 regions, but we were very interested in the overall trends. But one of the other things that we found in this analysis was that there are very beach-specific effects. There's sort of no one-size-fits-all for some regions. For Niagara, we found that there are very unique contamination sources in Toronto as well, the factory E. coli level. So you can't necessarily look at each region as a whole. Sometimes you need to look at it at a beach-specific level. We also published these results if anyone's interested, and I'll give you our website at the end of the presentation if you ever wanted to peruse and see what we've been up to. But we published those results in the International Journal of Environmental Health Research and Public Health. And I'll hand over to Binyam now.
Thanks, Johanna, for the nice introduction to our multiple studies. So now I will start with or continue with the other sites, the other 2 sites, Manitoba and Vancouver. So in Manitoba, still freshwater lake Winnipeg, and we included two beaches, which are located on opposite sides, Gimli Beach and Grand Beach. So we merged, so since they're like located close by, Grand Beach and Grand Beach East and West, we merged them. And we also included, or took information from one buoy, one weather station, one water level station to conduct this study. So, based on the data that has been collected from 2007 to 2021 in the two beaches, as you can see across the years, the yearly average daily geometric mean of E. coli seems to be higher, or is higher in Gimli Beach than Grand Beach across the years, you can see here. And also, we tried to see the exceedances, the days that the E. coli level exceeded the guideline amount, the guideline threshold amount, which is 200 CFU per 100 ml. And as you can see again here, that it's again most exceedances happened on Gimli Beach across the years than in Grand Beach. So, like Johanna indicated earlier, we try to link what we obtained from the public health data or the E. coli counts from the public health, and we linked it with publicly available environmental data. And so these are the variables that we used for this specific analysis from multiple sources, and as well like the 24-hour E. coli or the previous day E. coli. So to analyze this specific study, we built a mixed effect modeling where year for this particular study, we put it as a random effect because of the clustering of the E. coli amounts or the variability as you saw in the graphs across the years. So in the analysis with the intra-class coefficient estimates, they confirmed that the values or the E. coli counts clusters at the year level. And in order to select which variables should go into our model, we ran path analysis using direct cyclic graph, and we tested them in order to see which variable have some interactions or relationship in their effect on the E. coli counts. So based on that, we selected our variables, and we excluded the immediate variable which we found, which doesn't really need any adjustment, which is water level. So we conducted the analysis separately per each beach for the two beaches. And so what we found is a positive or an increasing relationship between 48-hour precipitation and 24-hour mean air temperature and the 24-hour previous day E. coli concentration. So it means when they increase, the E. coli concentration would increase. That's what we found for Gimli Beach. But for Grand Beach, we also found on top of these three variables, the 24-hour UV index, we found it to be having a negative effect on the E. coli count, which Johanna also explained, the negative effect because of its bactericidal effect on the E. coli, and also antecedent dry days, which are the days where there was no rain. So the more days without rain on the previous days would end up in resulting in like a reduced number of E. coli counts in the water bodies. And as I indicated earlier, year specific effects were seen, and like it suggests that yearly varying factors are affecting E. coli at varying levels. So we also tried to see the marginal effects of each predictor variable, which we found to be significant in our mixed effect logistic regression models. And we tried to see within that model and like adjusting for the other factors, how extreme values of certain environmental variables would affect the E. coli threshold exceedances. So like you see here, for 4 different values at the minimum, median, 95th percentile and 99th percentile values of rain, as you can see in the graph here. And we tried to see how rain affects based on the predictive probability of E. coli threshold exceedances. And you can see in the graph that the higher the rain, the 48 out precipitation amount, for example, 99 percentile 45.8, you will see like that the line on the top is the one that goes with it, and it shows that the higher the rainfall amount, the higher the exceedance would be for particular days across the years. So this is for rain, and similarly we did the same thing for mean air temperature. And the same thing, the higher the mean air temperature, the higher the E. coli threshold exceedances would occur. That's what it shows. And then it's suggesting that like extreme weather events might have extreme effects on the E. coli threshold exceedances. And like for the other beach, that one was for the Gimli Beach, and for Grand Beach, we also saw that again the mean air temperature, and like we found the same result, like higher result. And the shaded area you are seeing here is indicating the predictive interval. And similarly, again, we found out that like lower or minimum UV, it means when there was no UV and there is no bactericidal effect. So again, we expect the E. coli to be exceeding. So based on this, we concluded that we need to enhance beach monitoring programs for extreme weather events, as we saw in the marginal effects plotting. And we will use this finding to develop real-time recreational water quality predictive models, and also it can be used by others. So this paper is under review for publication. This result. And moving on to the Vancouver site, so we included 15 beaches on Vancouver. As you can see in the graph, they're dispersed across the beaches on mainly the Pacific Ocean, and we extracted data from multiple sources for this study. And similarly, when we look at daily geometric mean of E. coli yearly average, one beach which is not recommended for swimming, East Falls Creek, is showing a very extreme amount of yearly average geometric E. coli mean, and you'd see how its effect on the modeling too. You'd see it in my presentation. So like I indicated earlier, we used publicly available environmental data, and then data we obtained from the Vancouver Coastal Hills and Metro Vancouver, where we linked the data sources together to come up to see how combination of certain environmental factors or variables affect the E. coli outcome. So for this one, we use the Bayesian approach, mixed effect regression modeling, which functions through posterior prediction, and also it allows of making probability statements. I'm not going to dive into the detail of it, but this paper is accepted for publication in Epidemiology and Infection, so soon it will be out. So you might search and find it so that you can read more on it. So for this as well, we used a direct cyclic graph based on previous literature to determine which variables best predicts the E. coli on saline water, and we opted to feed a varying slope, which means varying predictor variable per each beach, so that like we can see how each variable have like a unique effect at each beach, so that we can have a more understanding of the association. And except for antecedent dry days, we couldn't find any significance of fitting it as a varying slope model, and so we excluded it and kept year as a fixed effect. And also we tried to see whether there was like some sort of interaction between some of the variables, and we found out, like, which is supported by the literature, the interactive effect of temperature and UV on the E. coli outcome in marine water. So this is our DAG, direct acyclic graph, showing the association between the variables. And again, since it was indicated that water level has a mediator effect, we don't need to control it. So we removed it from our model, and this is what you used. And so this is how it looks like, the overall relationship direction. So mean salinity has a negative effect. So it means the more saline the water, the less the E. coli count, which goes with the literature again, and rainfall in the previous 48 hours and previous sample day E. coli, because the sampling is not daily basis. So we use how previous sample day would have an effect on like the E. coli counts, the current E. coli counts, and we found a positive association, and as well for 24-hour temperature and UV index. So I'll explain this more, how the interaction is showing by using graphs for that you can easily grasp the message. So the average effects of the predictive variables it was indicated in the previous studies to showed like a high variation based on the model outcome. So here what you are seeing is the prediction, the posterior prediction plot, and it's showing how the predictive E. coli geometric value, or like the mean, is acting according to the previous sample day E. coli geometric mean, and we transformed the variable to a logarithm so that we would have a better understanding. So you can see that like there is a slight increase whenever there is an increase in the previous sample day E. coli geometric mean, there is an increase in the geometric mean, or the E. coli counts. So this is what I mean by beach specific effects. So you can see that there is like a difference in the effects across multiple beaches. For example, if you see the effect of previous sample day E. coli geometric mean on the E. coli counts, predictive E. coli counts, there is a strong association in False Creek Beach, and you would see that like for example in Castellano Beach, or like Rake Beach for sure, Locarno Beach, there was no or there is like a small effect than what we see in False Creek. So this is a very important finding. And when we see precipitation in previous 48 hours as well, there is an increased association, a positive association with the geometric E. coli count. And when you look at the beach specific effects, again, you can see it varies across the beaches, but the more rainfall predicts the more geometric E. coli. I mean, and salinity, as I said earlier, it has a negative effect. So the more saline the water, the less the predicted E. coli counts. And this also holds true for the beach-specific models. So you can again see mainly in False Creek, and then like if you see Rake Beach, like there is an effect, and like maybe like no effect on Castellano Beach. And moving forward, this is where the interaction comes. So here you can see that at the minimum value of UV index, there is a negative effect of 24-hour mean air temperature on the E. coli counts. But when you move forward on the median or 95th percentile UV index values, the temperature would have a positive effect, and there are certain or some explanations for this, like the type of, or like the strains of E. coli entering to marine water, which would make the E. coli strains to kind of develop some sort of protective mechanisms that would allow it to protect itself from the UV index, okay, the UV light, and then that would enable the survival of the E. coli in the water bodies. And For the minimum, there could be some other factors that like turbidity or wave height and things like that, according to the literature, that might have brought up this effect. So this is also shown showed like different effects across the beach, and this is just for the median value of UV index showing. So like we would have like 3 different graphs like this to show what it looks like and the minimum and 95% value of UV index to see how the mean air temperature affects the geometric E. coli count. So based on this, again, it has been indicated in our other studies that this kind of work should be focused on targeting beach-specific approach or beach-specific monitoring programs, and as well even predictive models is Vancouver in Vancouver or other marine water bodies. And I will pass on this to Johanna to tell you more about our real-time predictive models work. And this is what we found in the 2 sites. Johanna, over to you.
Thank you, Binyam. Okay, so I will continue here and then wrap us up. So this takes us into our second objective, so as Binyam said, was to develop the predictive models. And as I mentioned, we achieved this by using a type of AI modeling. It's called Bayesian network modeling. And we use the information that we learned in our objective 1. So we learned about our various regions, the environmental predictors that seemed to be particularly important in those regions, and we use that information to help us guide these. So while there is a big AI approach to it, where the computer essentially learns from the data that you give it, there is some manipulation from our side to direct the relationships of the variables, and our outcome variable, which was E. coli. And so we created these four network models were generated. And so you look at them and you just see arrows pointing everywhere. Well, what does that mean? Well, essentially, there were some general findings. We found that previous geometric mean, so that's the previous day, and that's currently what has been used at most public health units that don't have their own predictive modeling to make decisions about current day E. coli values. And the good news is that it is a very strong predictor, and it has strong individual predictive ability. Turbidity was very strong in Toronto and Niagara, and I touched upon that when I was presenting the Toronto and Niagara work. Salinity was very important in Vancouver, and temperature was particularly important in Niagara, Vancouver, and Manitoba. Rainfall was very important in Toronto, Niagara, and Vancouver, but did not have quite the same strong signals in Manitoba. So we're seeing some differences between the different public health units or regions. But again, the models, these AI models essentially use all the data that's available to them. So it's considering all the predictive models, but you can also, plug in and pretend, well, today I only have this data point. And so, as I mentioned, the geometric mean, the previous day geometric mean was a very strong on its own, even was able to quite predict with some accuracy, which is good news in terms of what public health units are doing. And so in terms of what we've been doing next with these predictive models is that we wanted to create a sort of web applications for public health units to use. We thought it'd be a nice place to have these predictive models live somewhere, essentially, where they could be accessed and use the models to actually create these calculations. So we developed an interactive web page for visualizing the beach and weather data, but also the home for our predictive models. And our goal for them is that they're user-friendly, that they're an informative tool for our public health units, and most importantly, that they're actually accurate. And so we've built this app now, and we have, this is just an example of a landing page for the Niagara region. And so you have a home page, and we're going to be testing them for summer 2024. And we're currently working with retroactive summer 2023 data to look at the predictive performance. So we're pretending we're in summer 2023, and we're actually going in and entering into our calculator. So this is what our calculator looks like. And so you enter the environmental data for your region. So for example, if you have yesterday, you have wave height. So this is all dependent on what the public health unit collects. If they collect wave height, so Niagara, for example, actually measures the wave height right at the shore. They know the previous day geometric mean. A lot of the public health units also measure water temperature when they're doing the sampling and turbidity. So you have that information, you plug it in. But we also have environmental data that's being automatically fetched. So that's yesterday's UV index. That comes from weather stations. We have our wind speed, we have rainfall, and then we have just the average air temperature. And so all that's automatically uploaded into, and then the health unit enters current day data, and that calculates a probability. So it tells you that the probability of the E. coli exceeding the threshold currently set by the 2012 guidelines. In this case, when we've entered some mock data, the probability is 31.58%. And so the public health unit would then take this information and make a decision. Okay, how comfortable are we with this 31%? You know, it's pretty low probability. I think we're okay to just continue with beach. But once you get to a certain threshold, you know, and each public health unit would probably set their own threshold. Like at what point might you want to evaluate the safety of water conditions for the day, and whether it's consistent to perhaps the current monitoring practice. You know, would you have made this decision based on just the previous day geometric mean, or now that you have this probability, would that change perhaps that decision? And so we're hoping that this tool, if it turns out to be accurate, and we're seeing, you know, some good performance happening already from a technical perspective, it would be nice. It would be nice to put into practice. But if it is useful, we hope it'll help beach managers in their decision making in terms of risk to bathers and people accessing beaches. In terms of other stuff we're working on some other pretty exciting projects that are very, very briefly go through now that we're going to be wrapping up soon, is the Canadian Beach Cohort Study. So in 2022, we conducted a feasibility study in the most popular beach in Toronto, and we collected data on beachgoers, and our goal here is essentially to understand the exposure to beach waters and the risk to acute gastrointestinal illness. We received bridge funding in 2023 to continue with our research, and we were able to use those funds to then do 2 additional beaches in Toronto, and we were able to enroll 623 participants from 405 households. And we surveyed participants and conducted follow-up to determine AGI. We followed them up within a week to see if they had been ill and if they had any other exposures to recreational water. And we also conducted additional water sampling on the recruitment days to do microbial source tracking. We were very excited to find out last month that we received funding from CIHR to do the full study. So we'll add beaches in Vancouver, Manitoba, and potentially other sites. We're hoping to do some in the East Coast as well, and further expand our research and recruit people from across the country that are attending beaches. So stay tuned for more on that. We're pretty excited about this work. We also received funding from the Public Health Agency of Canada to look at algal blooms. So this is going to be a similar study, and in fact, we're going to kind of sort of conduct a lot of it at the same time, where we're going to look at the burden of recreational water illness due to cyanobacteria and their toxin in Canadian freshwater beaches. We've received four years of funding for that. And we're going to examine the impacts of algal blooms on human and animal health. So we're going to look at people attending beaches, but we're also going to recruit people who have pets with them. So pet dogs. We've already conducted focus groups for both the beach cohort study and our algal blooms project. So we're going to understand hopefully a lot more in terms of exposures to E. coli and also to cyanobacteria and the toxins and how that has an impact on human health. So stay tuned for these exciting projects that we're really happy about getting funding for. And so in conclusion, we've applied different and novel analytic methods to understand the relationship of environmental factors of water quality at beaches across 4 regions in Canada. That was our first objective. And we learned which factors had an important effect, and specifically at the regional level and sometimes the beach level. We use this knowledge gained to develop predictive models using a very novel approach, Bayesian network models, which is an application of AI. And then we're actively exploring their health of burden and poor water quality levels and in algal blooms, and then further with our beach cohort study. We hope that these prediction models can be applied to global health context as well to understand water quality. And a web application like our Shiny can provide public health units with a tool for predicting water quality. So it's really our hope that we're actually able to give something to the public health units that may be useful in their risk management decisions. Information that's gained from our beach cohort and algal bloom projects will also have a wide spread impact in the area. We're very excited about sort of being Canadian first here. There is data, there are projects in these areas, but very limited in the Canadian setting. So we're very thankful to have this funding, federal funding, to be able to look at the Canadian context. And so I acknowledge, of course, our co-authors and our collaborators, our fantastic collaborators, which give us access to data and their knowledge. And here we have the leads from the various sites and all our partners across Canada, as well as our funding agencies. If you want more information, you're just interested to see what we're up to, you can visit us at canadianbeachwater.ca. Thank you very much.